probably all seen the magazine articles and the advertisements, the ones claiming to have the next new way to reduce the signs of aging, or maybe even the ones that are detailing the latest celebrities' facelifts or Botox treatments. But it seems like whether they're trying out a new healthy lifestyle trick they found in a magazine or taking a little trip to their local plastic surgeon, everyone has the same goal in mind, right? To try to find this so-called fountain of youth, or this way where they can stay feeling young and looking young for the entirety of their lives. Well, my name's Tejoni and I too am interested in finding this fountain of youth. So I'm working in the lab of Denise Montel, undergraduate student Dominique Houston, to better understand this process of aging by attempting to uncover the molecular mechanisms that are controlling it. In other words, we're trying to find genes that are involved in this process of aging, and we're doing this by looking at the Drosophila melanogaster fruit fly. And with the fly, we're looking at drug testing, so we're testing drugs that we've found to increase aging or increase lifespan. And then we're looking at a trait that the flies have that allows them to delay aging. And so the Drosophila melanogaster is a popular model used in research as a substitute for humans. This is because they share a lot of similarities genetically and physically with us. But unlike us, their entire genome or their complete set of genes and genetic material is mapped and known, which benefits us because gaining more knowledge on specific genes in the fly could help us gain more knowledge on specific genes in us. And so the flies that we're using come from the Drosophila Genetic Reference Panel, which is a population of flies that contains 200 fully sequenced inbred lines, and fully sequenced just means that their genome is known. And so they all come from the same genetic background. So in the same way that you and your brothers and sisters come from the same genetic background and are genetically similar and more genetically similar than you are to a stranger, these flies are also genetically similar. So you can almost think of them as like 200 brother and sister lines. And so because their genome is completely known, we can perform a Genomewide association study, or GWAS for short. And so this basically just means that we're going to be examining genetic variants between our fly lines. And to do that, we're doing this to find genetic variants that are linked with our specific trait. And so the genetic variant is pictured right here. So basically it's just a difference in DNA. So you can see the top one has like it has a G and a C right here, whereas this one has an A and a T down here. So those are differences in genetic variants. And so we can think of GWAS as like an input-output system. So the input we're going to put in is some data that's related to our trait, and then we're going to put it through the GWAS, and it'll come out with genetic variants that are linked with our trait. So the trait we're looking at for the first part is called Dipod, and it's a trait that flies have that allows them to delay aging in harsh environments. So you can think of it as like a program that's initiated when they're in an unfavorable environment, like a harsh winter, and it delays their metabolism and their locomotion and their cellular and organismal deterioration. So to look at the Dipod trait, we have to initiate it. We'll do this by simulating like a cold winter by putting in a 10-degree fridge. And then on day 35, we're going to take them out. We'll put them in 25 degrees where they age normally, and they'll stay in there until they live out the rest of their lives. And we'll test them against a control group that just stays at 25 where they age normally for their entire lives. And we're looking for the differences between good, bad, and non-diposers. The non-diposers would be the control group. And so we'll do this by looking at the post-dipos lifespan compared to the regular lifespan. And so what makes a good diposer is that they'll undergo the first 35 days, the entire dipos process, and then after that they'll come out and they'll look the length of their regular lifespan or longer than their regular lifespan. So they'll go through the first 35 days and they'll come out like the whole first 35 days never happened. And so for a good diposer, that would meet the change in lifespan, which is the difference between post-dipos and regular lifespan positive, because the post-dipos lifespan is either greater than or very close to the regular lifespan. And so you can see the data for a change in lifespan for all of our flies here. Each of these little boxes is a different fly line, and you can see the change in lifespan in days, going from positive and highest to decreasing. And so as you can see, only a handful of our flies are actually good diposers with a positive change in lifespan, and they can be seen in yellow. And then the rest of them just don't really dipose well. And so you can see the lifespan analysis for a good diposer here. This is just one of our fly lines, line 138. And it's, for the graph, it's days versus percent alive. So you can see the percent alive for the certain days for both the dipos in orange and control. And as you can see, there's a higher percent alive for the dipods on later days, meaning that overall they had a longer lifespan when they underwent dipods, making Lime 138 a good diposer. And so as I said before, we're going to perform a GWAS study. And so the data we're going to input is our change in lifespan, and we're going to put it into the GWAS. But when we did that, we got 80 genetic variants out, which is a lot to test for each one. So what we did was we looked at the genetic variants and the location of them in the DNA. And we were able to limit them to 5 genes for further analysis. So this is all of our preliminary data. And so what we're doing now is we're taking these 5 genes, and we're going to examine them using RTQPCR, which stands for Reverse Transcriptase Quantitative Polymerase Chain Reaction, which sounds really complicated, but it's just a way of looking at the DNA and searching for a specific sequence in the DNA encoding for a gene. And so we could use this to look at the gene expression of our 5 genes. And so we'll be doing this for both our diaposing flies and our control flies. And so we'll be sacrificing flies at different times in our process, so day 1, day 3, week 3, week 5, and week 7, and maybe more depending on how long they live for. And so we're going to do this to look at the gene expression of our 5 genes at various stages of the diapause and control process. And so here's a little overview of RT-qPCR. It can be a little complicated. And so basically we all have DNA, right? And usually for normal protein production, it goes from DNA, and then through transcription, it translates into RNA. Then through translation, we go from RNA to polypeptide or functional protein. But for reverse transcriptase quantitative PCR, it switches it up a little. So instead we do mRNA to complementary DNA instead. And we'll do this via reverse transcriptase enzymes. So we just switch around the process a little. And when we come out with this, we get a library of cDNA, and cDNA is just complementary DNA. And so with this cDNA, we amplify it, which just means we make a bunch of different replications of it. And then what we'll do is we put on a little probe, that's a fluorescence probe. You can see right here. And so the probe will, since it's a fluorescence probe, it'll allow us to see or count the amount of mRNA or the mRNA levels that are in our cDNA. And the mRNA levels correspond to the gene expression. So higher mRNA levels would mean the gene is expressed more. And so that'll help us look at our 5 different genes that we found. And then the second part of our project is drug testing. And so we're testing 4 compounds that have been found to increase lifespan of the flies. Not of the flies, of C. elegans in the lab of Michael Petrashek. And C. elegans is this little nematode right here. And so he created 4 compounds, and he's allowing us to test them on our Drosophila melanogaster fruit flies. And so we're going to try to look at how they work molecularly by testing them on 2 of our fly lines. One fly line has been found to have a shorter lifespan, and the other one has been found to have a longer lifespan. And so we're going to be testing these 2 because we want to see if it affects them differently. So do the shorter lifespan flies get a better, or do they get more benefits from it? Do they have more room to grow? Or is it just the same? And so we're testing these 2 fly lines using 6 different groups. The first one is our negative control, which will have no drugs. And the second is our group that's going to be tested with rapamycin. And rapamycin is a drug that's been found to increase lifespan, so that's our positive control. Then each of the 4 drugs will be tested separately. And so after we finish this, for our future work, we want to also perform a GWAS on these flies as well, similar to the one with dipods. So we're going to put in the change in lifespan. For this one, it'll be the difference between the drugs, or the flies that had the drugs and those that didn't. And so that'll go through the GWAS, and we'll get genetic variants out that we could then compare with our genetic variants that we got from the diaposing flies, in the hopes of finding genes that are involved in the aging process. And so understanding aging in fruit flies can better help us with understanding aging in us. And so as we age, we deteriorate, we become more susceptible to age-related diseases like Alzheimer's or other neurodegenerative diseases, or heart disease, or cancer. And so gaining more knowledge on this process of aging and the genes associated with it could better help us understand how to combat aging and these age-related diseases, thus bringing us another step closer to finding the fountain of youth. So I'd like to thank my lab, the Denise Motel lab, and everyone in it, especially my mentor, Dominique Houston, for answering all my questions and helping me become a better researcher. And I'd also like to thank Michael Petroshek for being our collaborator on our drug screen project. Then also EUREKA and CSEC for funding me this summer.